Good afternoon, everyone. We're gonna start in 5 minutes so that people can come and join in. Okay. Yes. So I guess we can start our class now, right? As you know, so today this is our last class, and I'm gonna take a big challenge. So what I'm going to do is, so there is one particular topic, I guess hypersensitivity, which I have not discussed with you guys. But as this is our last, so I want again same thing which we have done in the past 12 weeks is to revise, see things from the broader perspective. But this particular revision, it will be a little bit more faster, so that we can give more time to questions as well, right? So I guess we have more than 15-20 questions to get through. So what I'll do is, very quickly I'm gonna like skip through the slides, because obviously you are not familiar with these slides, right? Topics, and then we'll end up into hypersensitivity, and then we'll try to solve questions which are related to hypersensitivity, as well as then we are going to try to solve and see some questions which are related to vaccination, and then from those questions only we are going to understand the vaccination. Okay, so let me start. Okay, it is stuck, my pointer is stuck, just give me 1 minute. Huh. Okay, so first we studied about the immune system, right? So immunity usually refers to protection against infectious pathogens. Now all these things will be a little bit easier for you, right? In the first class, if you remember yourself, even this slide was overwhelming, that oh, what are these terminologies, what are these things. Now things are into its, like, you have the components and you can fit into the picture, right? So the collection of cells, tissues and molecules that mediate these reactions is called immune system, and the coordinated response of these cells and molecules to pathogen and other substances make up the immune response. Now you know why we are calling it a coordinated response, right? Because at every single step there is crosstalk, there is communication, there is sequential events which needs to take place to call it as an immune response, right? Then we saw the importance of immune system, so it's defense against infections, which we saw in majority of our class. Defense against tumors, it is also part of against infection, but as well as you can put this in um uh, what to say vaccination as well, or the basic principle of vaccinations. Control of tissue regeneration and scarring, cell injury and pathologic inflammation, recognition of injury to tissue grafts and newly introduced proteins. Then we saw what basically is infection and immunity. So stimulating immune responses against microbes through vaccination is the most effective method for protecting individuals against infections. Now you know what why we are saying this, right? If you already have a memory of things, the infection won't infect you or affect you to the extent it would have if you never had any memory, right? And the whole process of vaccination is that a memory should be there within you, so that if the pathogen enters at the second time, your symptoms will be less severe, your recovery will be faster, and all these things can be achieved by proper vaccination. So this approach has led to the worldwide eradication of smallpox. Okay, let me change. Okay, so these are the effectiveness of the vaccines, so we have gone through, and I don't need to go back to the previous numbers, this we all witnessed, right? So I don't have to go back like decades before, which we already have faced. And then we started with innate and adaptive, and you know these are just way of dividing things, right? So innate immunity is also called natural or native immunity, while adaptive immunity is also called specific and acquired immunity. So these terms, if you remember, you will understand better what active passive immunization is, passive immunization, active immunization. All those terminologies are related to the way or the branch of immunity they are taking help of. Then we saw the components of innate and adaptive immunity. Here you know that in innate immunity, you have physiological barriers, you have cells which are involved, which are just waiting and excuse me, they're just waiting for the microbe to come and they are not specific, right? Then we have adaptive immunity, where there is stimulation needed by the innate immune cells to let the adaptive arm of the immunity know that there is a pathogen, and then these things will be more specific and more targeted, right? And the innate immunity includes your epithelial barriers. Second line includes your phagocytes, plasma proteins, and now you know what which plasma protein we are talking about. It is the complement system, right? And then there are some additional roles, which is like triggering adaptive immune response, so that they both can work simultaneously to eradicate the infection. If we look at adaptive immunity, so these are the ones when innate immunity is taken care of, but if the sheer number is too much, or the type of infection is that it requires specificity, then adaptive immunity will be engaged, right? Then if we talk about adaptive immunity itself, it is divided into two parts: the humoral immunity, which is like basically things which are in the blood. Okay, so complement is in the blood, antibodies are in the blood, so it will be part of humoral immunity. When we talk about cell mediated immunity, it directly means T lymphocytes, right? So in this you have T helper cells, you have cytotoxic T cells, you have T regulator cells. All these cells, if you categorize them, they'll fit into cell mediated immunity. Now, humoral immunity is mediated by proteins called antibodies, which are produced by cells. So these are basically what? These are B cell receptors. Antibodies defend against microbes present in these locations by preventing them from infecting tissue cells and by neutralizing toxins. Okay, and antibodies also provide immunity to the newborn kid. If we talk about cell mediated immunity, so cell mediated immunity basically defends against microbes that have entered the host cell. Now humoral was in the blood, and when the microbes are inside the cell, then it is part of cell mediated immunity. Here also you can use your enhanced knowledge of immunology now, that even within the cell there will be intracellular microbes which are in the cytoplasm or in vesicles, and how they'll be targeted. Okay, if we look at the adaptive immunity and what is adaptive immunity, so it is developed after exposure to a microbe or its antigens. Okay, what is passive immunity? Passive immunity, here antibodies are transferred from an immune individual or produced in the labs. So if I am saying the immunity is type of active immunity, which means I am giving you the microbe or a part of microbe, or a protein or a part of protein, or say RNA which might make the protein. So these protein, the common thing will be that they are antigenic in nature, right? So that they can elicit an immune response. And after this exposure, once you have memory cells, once you have B cell, T cells specifically for this, it will be called as active immunity. Now if I am taking these antibodies and I am giving it to the other person whose body has not seen the microbe, whose body has not seen the protein, whose body has not seen the part of the protein and all those things, this will be called as passive immunity, right? If we look at the properties of adaptive immunity, so it is highly specific, diversity is there, major feature is of the existence of a memory, and then the best thing, non-reactivity to self. And if you are following the Nobel Prize, you should know that this year's Nobel Prize in medicine and physiology has been given to the major branch of immunology itself, right? So if you remember, we talked very less and very little about Treg cells, that is T regulatory cells. What were its functions? The functions of Treg cell was to basically calm down the activated immune response. So microbe has entered, we know what happens when the microbe has entered, right? But we know very less about when microbe has been cleared successfully, what happens after that, right? So that part is where there are specialized cells which are involved, and the discovery of those cells have been awarded the Nobel Prize this year. Okay, then clonal selection comes into play. You know very well what clonal selection is, because at every single point you have n number of choices to make, so you have to be very very selective to provide the specificity or the specific nature of the adaptive immune response, and clonal selection is one such mechanism through which specificity is maintained. Other features include adaptive immune responses have other characteristics that are important. Immune responses are specialized, different responses are designed to defend best against different kinds of microbes. All immune responses are self-limited and decline as the infection is eliminated, allowing the system to return to the resting state, prepare to respond another infection. So this part, it is just single statement, right? But how is it done, which components of the cells or the system is involved, so that discovery is still going on. Okay, then the cells of the adaptive immunity includes lymphocytes. In lymphocytes, it will be B lymphocyte, T lymphocyte. Now you know, now you have a picture of these cells, right? Earlier when I was introducing these cells, this was just an information to you, right? And then we talked about myeloid cells. So then you know the function of neutrophils now, macrophages, dendritic cells, and mast cells as well. This is the graph which I mentioned earlier, that this is the basis of immunology or immunity, and this is the same graph which we exploit. The understanding of this graph, basically, we exploit to design our vaccinations. Okay, so the primary and secondary immune response, I don't think so that I have to go deeper into this. You know, when the first incident will happen or the first infection will happen, it will take a little bit time for the body to get to know what infection is this, and it will develop an immune response. But again, if the similar antigen affects us, this time they will, without the delay in eliciting the immune response due to the presence of earlier memory, the response will be quick and peaky, like the concentration of the or the effect of the infection effect of the reaction of the body will be much more in comparison to when it was in the primary response, right? Then we saw the classes of lymphocytes. When we talk about B lymphocyte, T lymphocytes, what exactly are in the B lymphocyte and T lymphocyte? So these are the common cells which are there throughout the immunology, right? So B lymphocytes, helper T cells, cytotoxic T cells, T regulatory cells, and keep this in mind that these are not the only cells. So to introduce you to the immunology subject, or to introduce you to the major components playing role, this course was designed, right? So it's a very introductory preliminary course, so you can always dig deeper into what happens, like what sort of T helper cells are there, what are its receptors, what is their CD state, and all those things. Okay, then we talked about the specificity and receptors of innate immunity and adaptive immunity. So these features you are like very well aware of. Then we introduced the first ever response, like how our body is basically acting against the infection, which was the inflammation, right? And here now, with your existing knowledge, you can add the players, that is the complement as well. Okay, then we saw the events which are basically taking place during the infection, and how the inflammation is taking place, helping in like eradicating the infection. Then we saw the innate immune system, what receptors or what components of the cell, especially receptors, are responsible for eliciting this response. So they are present in membranes, like on the surface, as well as in the cytoplasm of the cell. Then we have discussed to extents that when we say, oh, cell knows that there is an infection, or cell needs to behave in this, or cell changes its behavior once it sees a pathogen organism, we understood what exactly it means, right? So it is basically signaling how the cell is seeing a ligand, here say microbe, and then acting accordingly. right? Then we started to look into the antigen recognition, how the adaptive system is actually seeing the antigen, because innate immunity, there are cells floating here and there, they can directly go and look at the microbe or search the microbe and just eat it, right, and digest it and clear it. But adaptive system needs to see it via MHC molecules, or the major histocompatibility complex cells having these receptors. So the receptors of lymphocytes include your immunoglobulins and your TCRs, or the T cell receptors. Then we dig little bit deeper into the major differences between T cell receptor and B cell receptor. Here you were introduced with the gene recombination, that VDJ joining, and all those things were part of it. I guess it will be nice to refresh this particular memory. Just give me 1 minute. So again, as I've mentioned, we are not going to go deeper into it, but surely revision has its role, and we are going to take the maximum out of our revision session.
Number 1. There are 2 branches to the immune system.
I
Ways.
just want T cell receptors.
In immunology, putting an end to an infection or bringing an infection under control is often referred to as containing an infection. While B cells and T cells are both components of the adaptive immune response, they contain infections in different ways. B cells produce and secrete antibodies. Antibodies are proteins that are able to bind to bacteria and viruses. Antibodies binding bacterial cells or viruses prevents infections from spreading. The antibodies block the bacteria from growing colonies, and in the case of viruses, the antibodies bind to the viruses and prevent the viruses from infecting cells. Any disease-causing agent, such as a virus, can be referred to as a pathogen. The process of antibodies binding a pathogen to contain the infection is known as opsonization. Opsonization by antibodies not only prevents the spread of infection, but marks the bacteria or viruses for destruction. Phagocytic cells circulating in the blood and fluid surrounding the tissues of the body can detect the presence of antibodies bound to pathogens and then engulf and destroy the opsonized bacteria or viruses. These phagocytic cells are not B cells or T cells and are part of the innate immune response. So while the process of opsonization is part of the adaptive immune response, the actual destruction of pathogens involves the innate immune response. Therefore, both branches of the immune system must work together for a complete immune response to infection. Further detail on this phagocytic process is provided in the video on this channel on inflammation. When a cell becomes infected and cell-mediated immunity is needed, T cells respond by killing the infected cell. There are numerous types of T cells. Cell-mediated immunity is largely dependent on a class of T cells known as CD8 cytotoxic T cells. The CD8 cytotoxic T cells are called cytotoxic because they are able to kill other cells. When a cell becomes infected with a virus, a CD8 cytotoxic T cell responds by killing the cell infected with the virus. Since viruses replicate inside of cells, killing the cell infected with the virus stops the ability of the virus to replicate. Additionally, while often not sufficient to rid the body of cancer, CD8 cytotoxic T cells are also able to kill cancer cells when they detect cells of the human body that have become cancerous. Thus, while CD8 cytotoxic T cells are killing cells of the human body, whether virus-infected or cancerous cells, this can actually preserve health overall in the body because the killing of the infected cells can stop the spread of the virus or cancer. Cell particles and viral proteins that are released into the fluids of the body after the infected cell is killed can then be destroyed by phagocytic cells, just as is the case with opsonization. To summarize adaptive immunity, B cells respond by releasing antibodies for opsonization of pathogens, resulting in the destruction of the pathogens. T cells respond to infected cells by killing the infected cells. From that infection are specific only to that one strain of the flu. If those same antibodies that stopped the spread of this one strain of the flu were then exposed to a common cold virus or a streptococcal strain of bacteria, The antibodies would not be able to bind and would not at all be effective in containing the infection. This is due to the specificity of the antibody binding site, the portion of the antibody that binds to a bacterial cell.
So this is not any further a new information for you guys, right? Now you can imagine, you can understand the chemistry behind it, you can understand the structural biology behind it, right? So then we discuss a little bit more on the antibodies, how they are made. So I'm gonna play this again because this holds an important lesson of recombination, and chances are, like, theoretically you have to be a little bit clear and understand the complexity of the process, how antibodies are made, because there are constant regions, variable regions, and only the variable region is the one which is identifying the antigen. So how beautifully, with the like probability of error and chances, we are able to get specificities. Okay.
We'll discuss B cell receptor. B cell antigen receptors are immunoglobulins expressed on the surface of B cells. The receptor is produced terminally differentiated by B cells or plasma B cell and secreted as antibodies into the blood. This is extracellular region of B cell. This is cell membrane. While this is cytoplasmic region of B cell. Immunoglobulins are the glycoproteins, composed of two identical heavy and light chain. The weight of heavy chain is 50,000 to 70,000 Dalton, while the weight of light chain is 25,000 Dalton. There are two types of light chains denoted by kappa and lambda. Cysteine residues form bridge between individual chains of immunoglobulin molecule. This is antigen binding fragment, or Fab, while it is crystallizable fragment, or FC fragment. They are separated by enzyme called as papain. FC fragment possessed binding sites for complement factor C1q. Light chains consists of two regions.
Can you guys answer why heavy chain weight is in a range? If yes, put your answers in the chat box. It's just the it's not a hypothetical question, but we have not covered this. But you can think in the direction of how many domains an antibody has. Okay.
Of equal size. This is variable region of light chain, subdenoted by VL. And this is variable region of heavy chain, so it is denoted by VH. This is constant region of light chain, so it is denoted by CL. And constant region of heavy chain, so denoted as CH, which is further divided, so denoted as CH1, CH2, and CH3. The constant region varies little from one immunoglobulin to another immunoglobulin. There is huge variability among amino acid sequence in variable region. Both constant and variable region consists of about 110 amino acids. Look at the heavy chain. It has one variable region with 110 amino acid and 3 constant regions. But in case of immunoglobulin M and immunoglobulin E, there are 4 constant domains. Don't worry about the structures of immunoglobulins. They are also explained in detail later in this video. But remember that immunoglobulin M and immunoglobulin E have 4 constant regions. Due to the variability among amino acids, in variable region, immunoglobulin variable portion varies from one immunoglobulin to another immunoglobulin and involve an antigen binding. This is N-terminal. This is C-terminal. Now, C-terminal of B cell receptor has spacer region having 26 amino acids. Then, this is transmembrane portion.
Correct. So you can always go back to these things as it is part of the presentation itself. Then we look deeper into the structure of antibodies. How many constant regions, how many variable regions, how exactly they look like. So this is a representative way, right? This is how exactly they will be in your system, and this is a structural say configuration and how they are represented in the structure form or secondary structure form. Then we looked at different types of antibodies. This is the very first time we saw that antibodies are of different types, how many chains or monomers they are made up of, what is their half-life, and from this, based on your now things or the information which you have, you can see that their half-life or serum concentration you can directly relate to with respect to the infection, right? So if the infection happens for the very first time, you know IgM will be more. When infection happens for the second time, then you know it will be IgG or IgE or IgA, depending upon the type of infection, right? And you also know and can see from the structure itself that why IgM will be the first antibody to be there. Okay, then we talked little bit about the monoclonal antibodies and how they are made by the use of hybridoma technology. So I have a dedicated video aid for that in the question section, I guess. So we'll discuss that at there itself. This is basically how the hybridomas or the monoclonal antibodies are made. So the question is, why do we need monoclonal antibodies in the first place? Why can't we have generic antibodies which are there and just proceed with it, right? So this amount of research, time, and protocol generation, and then setting up industries which are just making monoclonal antibodies, why it is important? Because monoclonal antibodies, you can get the say vial of monoclonal antibody, you know that they are going to bind only to the antigen against which these antibodies have been raised. There will be no cross reactivity. There are like higher chances and more probability that the causative agent or the antigen for which you are administering the antibody, it will be targeted because it is coming from monoclonal source and it is of monoclonal type. Okay, then we discussed about the TCRs or the T cell receptors. Basically we discussed about their structures just like we discussed about antibody structures. So both B cell receptor and T cell receptor, they are similar yet different. Okay, the major difference will be that T cell receptors, they are not secretory in nature, they have to be bound to the membrane, and the moment we say that they are membrane bound receptors, they should have a transmembrane domain or a membrane embedded domain associated with them. Okay, then we delve into the MHC, because you know without MHC TCRs are of no use. TCR recognizes the MHC which displays the processed antigen, and hence the adaptive immune system is activated. So we have to have proper understanding of which class of MHC binds to which class of TCR or which type of TCR. Then okay, so I'm going to go through it, it will be helpful for you guys. In the adaptive immune system, the molecules responsible for specific recognition of antigens are antibodies and T cell antigen receptors. Antibodies, also called immunoglobulins, can be produced as membrane receptors of B lymphocytes and as proteins secreted by antigen stimulated B cell that have differentiated into antibody secreting plasma cells. While TCRs or T cell receptors are membrane receptors and are not secretory in nature at all. The core structure of the antibody consists of two identical heavy chains and two identical light chains. The cell receptor consists of an alpha chain and a beta chain. Each chain contains one V region and one C region. The V region of immunoglobulin and TCR molecule contain hypervariable segments called CDRs. The inherited genes that encode antigen receptor consist of multiple segments separated in the germline and brought together during maturation of lymphocytes. Receptors of different specificities are generated in part by different combinations of V, D, and J gene segments. So this is all trial and error things. You tried one combination, it worked, the receptor worked, is able to recognize, that joining will be selected. On the other hand, you join, you made one receptor, it is not recognizing any antigen, the cell will be degraded, or the receptor will not be allowed to be present in the system. During their maturation, lymphocytes are selected to survive at several checkpoints. You know why we need them, because if there is any disturbance or any error in the checkpoints, it leads to diseases, right? Only cells with complete functional antigen receptors are preserved and expanded. In addition, T lymphocytes are positively selected to recognize peptide antigen displayed by self-MHC molecules. Immature lymphocytes that strongly recognize self antigens are negatively selected and prevented from completing their maturation, because we don't need unnecessary load, right? Then we looked into the T cell mediated immunity activation of T lymphocytes. So two ways through which T lymphocytes or T cells will be taking part or making the adaptive immune system. Once, when the microbe is inside a vesicle or a phagocytic vesicle or inside a phagocytic cell, and other in which the microbe is in the cell itself, and we need to clear that out. Then we looked into how the induction and effector phase of cell mediated immunity works. So this is the first time when you actually saw two different arms of the immunity, which we were talking since the beginning, how they come together and how they are joined together, and at the end the destination is always to clear the microbe, whether it is inside the cell, whether it is outside the cell, whether it is inside a phagocytic vesicles, or whether it is in the cytoplasm. Okay. Then we looked into the steps in the activation of T lymphocytes. So you know the first thing will be, for the active T cell, is to be paired with MHC cells, and the cells which have MHCs or which are presenting the antigen are called antigen presenting cells. Then a lot of change. Now you know the changes are what I mean by change, which means signaling. A lot of signaling will happen after the cell has recognized the antigen bound to the MHC molecule presented by the APC, leading to the decision whether to have like T helper cell or CD toxic T cell, or what steps to be taken care of. It will completely depend upon the type of MHC molecule the cell has recognized. Then we looked into, and another major component is that it is not only the MHC, we need co-receptors as well. If those co-receptors are not fulfilling the binding protocol, then there won't be any activation of the T cells. Then we looked into the antigen recognition and signal transduction during T cell activation. So whatever we have seen here in the big version, this is the focused and concentrated version. So we start to have conversation about only this section, this region. But see, there are 1, 2, 3, many different types of co-receptors are involved to actually have an effective activation of T cells. Then again, putting a little bit more focus on the signal transduction pathways, because signaling is the one which is basically taking the steps, right? So once the ligand has come, ligand has been bound to the receptor, after that every single thing will be governed by the cyclic reactions which are taken place in the cell. Majority of them can be seen here, but these are not the only ones. There are other signaling pathways as well which are activated once the ligand comes and binds to its receptor. Okay. Then the overall summary was that T lymphocytes are the mediators of the cell mediated arm of the adaptive immune responses, which combats microbes that are ingested by phagocytes and live within these cells or microbes. The response of T lymphocyte consists of sequential steps. You know what those steps are. And then the T cells use their antigen receptors to recognize peptide antigen. So here keyword is peptide antigen. T cells cannot see any other type of antigen, and you have all the information now. The information is MHC can only show small peptides, can only display small peptides. Okay, so antigen recognition by the T cell receptor triggers signals that are delivered to the interior of the cell by molecules associated with the TCRs. The binding of T cell to APC is enhanced by adhesion molecules, notably the integrins, whose affinity for their ligand is increased by antigen recognition. APCs exposed to microbes or to cytokines produce as part of the innate immune reactions to microbes express co-stimulations or co-stimulators that bind to the receptors on the T cell, and making sure that correct cell which has the pathogenic peptide displaying on it is recognized by the T cell. The biochemical signals triggered in the T cell by antigen recognition and co-stimulation result in the activation of various transcription factors, which are finally changing the transcriptomics of the T cell, and the effect is shown. The TCR signaling pathways involve protein tyrosine kinases, which phosphorylate proteins that become docking sites for additional kinases and other signaling molecules. In response to antigen recognition and co-stimulation, T cells secrete cytokines that include proliferation of the antigen stimulated T cells and mediate the effector function of T cells. T cells proliferate following activation by antigen and co-stimulators, resulting in expansion of the antigen specific clones, because we need more of such cells. Some of the T cells differentiate into effector cells that are responsible for eradicating infections. CD4 positive effector T cells produce cytokines and surface molecules such as CD40L to secrete different cytokines and do their function. Other activated T cells differentiate into memory cells, which survive even after the antigen is eliminated. Naive T cell migrate to peripheral lymphoid organs, mainly lymph nodes, draining sites of microbial entry, right? Because if they are not in blood, they are not in they are inside the cell, but we cannot keep these T cells and B cells which are being selected to always float in the blood, right? So there has to be like contingent areas in which these cells are densely populated. The pathways of migration of naive and effector T cells are controlled by adhesion molecules and chemokines. So if we look at the subset of T helper cell or different types of T helper cells, we have Th1, Th2, Th17 and TFH, that is follicular cells, follicular helper cells. So these are just the cells, again saying a lot of work is going on in this field, a lot of things are known at the same place, a lot of things are not known. Okay, so the information which you have, it's not an absolute information, the information is always getting added on, and in one context one information can be true and in another context that information might be false. Then we looked at the properties of the major cytokines. Again, you don't need to remember all these, like especially talking about with this slide, with respect to this slide, you don't have to remember every single thing, but majority of the things should be there. For example, IL-2, IL-17, interferon gamma, right? So these are some of the major interleukins which take place, and don't you worry that you have to like keep remembering them, you are not able to remember. The more you read about them, it will become very natural to you ki, oh, if there is an inflammation, which interleukin is high, if there is repair mechanism going on, which interleukin will be involved there. So they come intuitively once you get in touch with the respective literature. Then we looked at the cooperation between CD4 and CD8 T cells in eradicating the intracellular infection. You know, T helper cell, as the name suggests, they'll help in killing the cell which is infected, and cytotoxic T cell will directly kill the microbe which is infected, right? Then we looked at the migration of naive and effector T lymphocytes, how they are migrating, which type of receptors, which type of interactions are enhanced when the cell is moving, when the cell is being called at a place where infection is taking place. Then we looked into the evasion of cell-mediated immunity. Despite having all these measures, there are organisms who has learned or adapted themselves to escape these checkpoints or escape these policemen, right? So with our knowledge and education, we are actually using these techniques which nature has blessed apparently these microbes to come up with unique and different ways to protect ourselves. So, cell mediated immunity is the arm of adaptive immunity that eradicates infection by cell associated microbes. CD4 positive T cells can differentiate into subsets of effector cells that make different cytokines and perform different functions. Effector cells of the Th1 subset recognizes the antigen of microbe that have been ingested by the macrophages. Classically, activated macrophages produce substances including reactive oxygen species, NO species, and lysosomal enzymes. Th2 cells stimulate eosinophilic inflammation and trigger the alternative pathway of macrophage activation. The balance between activation of Th1 and Th2 cells determines the outcome of many infections, with Th1 cells promoting the Th2 cells suppressing defense against intracellular microbes. Th17 cells stimulate the recruitment of neutrophils, while CD8 positive T cells differentiate into CTLs that kill the infected cell mainly by inducing the apoptosis of the cell. Many pathogenic microbes have evolved mechanisms to resist cell mediated immunity. These mechanisms include inhibiting phagolysosomes, fusion, escaping from the vesicles of phagocytes, inhibiting the assembly of class 1 MHC peptide complexes, and producing inhibitory cytokines or decoy cytokine receptors. Then we had a look at the central and peripheral tolerance to self antigen. This is the topic which got the Nobel Prize this year in medicine and physiology. Okay, so there is central and peripheral tolerance. So what do we mean by that? So central tolerance, basically the immature lymphocytes specific for self antigen that encounter these antigens in the generative lymphoid organs may be deleted. B lymphocytes may change their specificity, and some T lymphocytes develop into regulatory T cells. Some self-reactive lymphocytes may complete their maturation and enter peripheral tissue. Peripheral tolerance indicates mature lymphocytes that recognize self-antigen may be suppressed by Treg inactivated or deleted. So the types of cells involved in tolerance was is still under a lot of debate how it happens, which components are playing major roles. So a lot of research is being done in this area, and this is again the similar thing which we discussed. Then we talked about the types of or the phases of humoral immune response. You know what we are talking about here? We are talking about the immunoglobulins, because humoral immunity will involve antibodies as well as complement system. This is what we saw. Then again, these are similar things which will be repeated a little bit and will be presented in different way. Then we discussed about the what I wanted to show you guys. So overall, what we understood about the humoral immunity was humoral immunity is mediated by antibodies that bind to extracellular microbes and their toxins. Humoral immune responses are initiated by recognition of the antigen by specific membrane Ig molecule or the immunoglobulin molecule. In humoral immune response to a protein antigen called T-dependent response, the binding of protein antigen to a specific Ig receptor of naive B cell in lymphoid follicle results in the generation of signals excuse me, that prepare the B cell for interaction. So with your information, you know that even the B cell will have multiple stages of activation. It will be double positive, it will be double negative, it will be positive, negative. So all those things are also there. The full T dependent humoral response develops in germinal centers and lead to extensive class switching and affinity maturations. So these are two such topics which are important and you should be pretty clear about them. So affinity maturation is the process by which the affinity of antibodies for protein antigen is increased with prolonged or repeated exposure to the antigens. The process is initiated by signals from T follicular helper cells, resulting in migration of the B cell into follicles and the formation of germinal center. Polysaccharides, lipids, and other non-protein antigens are called T-independent antigens because they induce antibody response without T cell help. Most T-independent antigens contain multiple identical epitopes that are able to cross-link many Ig receptors on a B cell, providing signals that stimulate B cell responses even in the absence of helper T cell activation. Secreted antibodies form immune complexes with residual antigen and shut off B cell activation by engaging an inhibitory Fc receptor on the B cell. This is the overall representation of how cellular events are taking place in the germinal center, how a naive B cell is there, how T cells are there, where exactly these clusters are present there, which cell is actually showing them the antigen, and how they are being selected and rejected at the same time. And then we learned about the effector functions of antibodies. So we have these different types of antibodies, but why do we have different antibodies? We can have one antibody which can function which can basically function for everything, but there are different functions and different antibody properties are suitable for these different functions. And what are these functions? These functions are neutralization, opsonization, antibody dependent cellular toxicity, phagocytosis, and inflammation. And these were the differences and categories between what sort of infection and what sort of location or the spatial distribution of the microbe require for its clearance. Then we looked at the antibody mediated opsonization. So in single word, if you want to describe opsonization, is that if you see antibody on any say surface of a cell, it automatically becomes visible to the system. So it's like I'm putting a flag on a cell. Now this flag will alert every thing which is surrounded by this, right? So opsonization means making it more palatable. So if antibody is there, phagocytosis will be easier. If antibody is tagged there, many other receptors can come and see that, oh, something not good with this cell, and stay away from these cells. So everything happens once you put a flag on it, and opsonization is exactly that. Then we entered into the complement system. So complement system, as I have mentioned even during when we were learning this, you have to know that where these pathways are meeting, what is the initiation factor or the initial complex which is made, and then you are sorted for the complement system. This is one of the easiest as well as the most typical concept in immunology. If you know it, you can very well clear the questions which are associated with the complement system. Okay, so try to make this understanding very clear in your head.
The system is composed of over 30 proteins, most of which are found in serum, while the remaining proteins are membrane-bound proteins and receptors. On activation, complement proteins that participate in the activation of either the classical or alternative pathways of complement interact in a highly specific enzymatic cascade and generate proteolytic fragments that mediate the numerous biological functions of the complement system. In this video, we will focus on the activation and biological functions of complement. Activation of the classical pathway. The classical pathway is typically activated by antigen-antibody complexes. Although there are many other activators of this pathway, only two isotypes of antibody, IgM and, as shown here, IgG, will activate the classical pathway. While one pentameric IgM antibody will activate the classical pathway, a minimum of two IgG antibodies in close proximity are required. In either case, antibody must be bound to antigen in order to activate the classical pathway. The classical pathway is activated when C1, the first protein in the pathway, binds to the Fc portion of the antibody. C1 is a large macromolecule composed of a C1q molecule and two molecules each of C1r and C1s. It's the globular head portion of the C1q that binds to the Fc binding site on the antibody. This binding causes a conformational change in C1q and subsequent autocatalytic conversion of C1r to an active serine protease. Activated C1r cleaves C1s to an active serine protease. Activation of C1 by binding antibody is also termed complement fixation. The next step in the activation of the classical pathway is the cleavage of C4 by the activated C1 molecule. C4 is cleaved by the activated C1s molecule in C1 into two proteolytic fragments, a small peptide called C4a and a larger fragment, C4b. C4b attaches covalently to the antigen-antibody complex and has a binding site for C2, the next protein cleaved in the classical pathway. C2 is also cleaved into two fragments, and the resulting C2a fragment binds to C4b. This bimolecular complex of C4b2a is a C3 convertase of the classical pathway and serves to cleave C3 through the enzymatic activity in the C2a portion of the complex. Classical pathway C3 convertase. The C3 molecule is cleaved by the C3 convertase into C3a and C3b. The C3a fragment has potent biological activities, which will be discussed shortly. The C3b fragment can covalently attach to the C3 convertase, resulting in a trimolecular complex consisting of C4b, 2a, 3b. This multimolecular complex is called the C5 convertase of the classical pathway and is specific for cleaving C5. Classical pathway C5 convertase. The C5 molecule is cleaved by the C5 convertase into C5a and C5b, both of which mediate important host defense functions of the complement system. Both C3a and C5a are potent chemoattractants. C3a attracts mast cells to sites of complement activation, and binding of C3a and C5a to these cells, as well as basophils, induces degranulation and release of histamine and other vasoactive amines. C5a is a chemoattractant for macrophages and neutrophils, and binding of C5a to these cells primes them for mediating their host defense functions. The terminal complement pathway, or formation of the membrane attack complex. One of the best-known host defense functions of the complement system is its ability to lyse many bacteria, envelope viruses, and nucleated cells. Lysis of cells by the complement system is mediated by a large macromolecular structure called the membrane attack complex, or MAC. The MAC is formed through what is called the terminal complement pathway, which starts with the generation of C5b. C5b associates with C6, C7, and C8, forming a large multimolecular complex that associates with and begins to disrupt the cell membrane. Subsequently, C9 binds to C8, and this is followed by the binding of many additional C9 molecules, leading to the formation of a large pore in the cell membrane. The pore in the membrane is large enough to allow water, ions, and small molecules to enter the cell, leading to lysis. Activation of the alternative pathway. The alternative pathway of complement is activated by LPS and other lipopolysaccharides found on the surface of invading pathogens. The alternative pathway is always active at a very low level. This activation is due to the spontaneous hydrolysis of an internal thiol ester bond in C3. Hydrolyzed C3, called C3 water, has a binding site for factor B, another protein in the alternative pathway. Factor B binds to C3 water and becomes a substrate for cleavage by factor D, denoted here as FD. Factor D cleaves factor B into two fragments, BA and BB. The enzymatically active BB fragment remains associated with C3 water, and this bimolecular complex is the alternative pathway C3 initiation convertase. Alternative pathway C3 initiation convertase. This initiation convertase cleaves C3 into two fragments, C3a and C3b. The newly generated C3b can covalently attach to nearby surfaces, just as C4b does in the classical pathway. In the absence of an appropriate activator for the alternative pathway, complement regulatory proteins block further activation of the pathway. However, C3b, on interacting with an activating surface, such as a bacterial cell, will bind factor B, which will be cleaved by factor D, resulting in the formation of the alternative pathway C3 convertase of C3b-BB. Alternative pathway C3 convertase. The C3 convertase of the alternative pathway cleaves C3 to C3a and C3b. Some of the C3b generated by this cleavage does not interact with the activating surface and remains soluble. Some of the C3b generated will covalently interact with the activating surface and start the formation of additional alternative pathway complexes, thus rapidly amplifying activation of complement. Complement through this pathway. The surface of an activator, such as a bacterial cell, can rapidly be covered with many thousands of C3b molecules in a few minutes as a result of this amplifying effect. And, as with the classical pathway, some of the C3b generated will interact with the C3 convertase itself, resulting in the formation of a trimolecular complex of two C3b molecules and BB. This multimolecular complex is called the C5 convertase of the alternative pathway and is specific for cleaving C5. The C3a and C5a fragments generated by activation of the alternative pathway mediate the same functions of chemoattracting and activating granulocytes and phagocytic cells. C3a attracts mast cells to sites of complement activation, and binding of C3a and C5a to these cells, as well as basophils, induces degranulation and release of histamine and other vasoactive amines. C5a is a chemoattractant for macrophages and neutrophils, and binding of C5a to these cells primes them for mediating their host defense functions. Activation of the alternative pathway results in the formation of the membrane attack complex, leading to the lysis of the invading pathogen. Lysis of cells by the complement system is mediated by a large macromolecular structure called the membrane attack complex, or MAC. The MAC is formed through what is called the terminal complement pathway, which starts with the generation of C5b. C5b associates with C6, C7, and C8, forming a large multimolecular complex that associates with and begins to disrupt the cell membrane. Subsequently, C9 binds to C8, and this is followed by the binding of many additional C9 molecules, leading to the formation of a large pore in the cell membrane. The pore in the membrane is large enough to allow water, ions, and small molecules to enter the cell, leading to lysis.
Yeah, so that was the whole complement system. So we are not going to go into the theory of it. Yeah, so here's the overall representation of three pathways: alternative pathway, classical pathway, and lectin pathway. So what I mentioned, what you have to remember is what is C3 convertases, like the initial building block. what is C5 convertases, these two components of all these three pathways, and what is the recognition thing or the initiation of these pathways, because the name of these pathways dependent upon what they are recognizing or how they are being initiated. Okay, then different functions were enlisted. Then yeah, so at the end of the day, when the MAC is being made or the membrane attack complex is being made, you don't need to remember specific concentration in this, but you should know that MAC is made up of how many components. So it is made up of C5, C6, 7, 8 and multiple units of 9. Okay, and again, these complement system also help in eradicating the infection by opsonization, by stimulating the inflammation, and then by cytolysis, that is making of the MAC complex on the cell itself. This is the overall representation of that, only this part. So this we have not discussed. Okay, so we are going to go a little bit faster to this because we can understand these topics from the questions and the previous year questions as well. So the concept that the immune system is required for defending the host against infections has been emphasized throughout the book. The book which I am referring here is the same book which is discussed in the lectures. Okay, then no, I guess we have discussed this. Okay, yes, we have discussed this, so we're gonna go a little bit faster in this. So there are basically 4 different types of hypersensitivities, that is immediate hypersensitivity, antibody or type 2 antibody mediated or type 2 hypersensitivity, type 3 hypersensitivity and type 4 hypersensitivity. So what is the difference between these type of hypersensitivities is basically again the same thing. How exactly is the reaction coming from? Is it the reaction because of the antigen antibody interaction, or is it the reaction which is coming when the antibody has recognized the antigen and then it is overstimulating the inflammatory pathway, right? So all those things are enlisted here. Similar thing was there, I think we have discussed this. Yeah, so we're gonna go into this also we have discussed clinical symptoms as well. So we can just see the summary. So immune responses that cause tissue injury are called hypersensitivity reactions, and the disease caused by these reactions are called hypersensitive diseases. Hypersensitivity reactions may arise from uncontrolled or abnormal responses to foreign antigens or autoimmune responses against self-antigen. Hypersensitivity reactions are classified according to the mechanism of the tissue injury. According to the mechanism of the tissue injury. Immediate hypersensitivity, type 1, commonly called allergy, which we say it's a seasonal allergy or pollen allergy, those things, is caused by the activation of T helper 2 or Th2 cells and interleukin 4. Here you should be specific with respect to the interleukins. Okay, and interleukin 13D producing follicular T helper cells, and production of immunoglobulin E, IgE, antibody against environment. Clinicopathologic manifestation of immediate hypersensitivity results from the action of mediators secreted by the mast cells, that is amines increase vascular permeability of and dilate blood vessels. Arachidonic acid metabolites cause bronchial smooth muscle contraction, and cytokines induce inflammation, the hallmark of the late phase reaction. Treatment of allergies is designed to inhibit the production of mediator, antagonize their action, and counteract their effects on end organs. Antibodies against cells and tissues antigen may cause tissue injury and disease, such as type 2 hypersensitivity. IgM and IgG antibodies activate complement, which promote phagocytosis. The immune complex disease type 3 hypersensitivity, antibodies may bind to circulatory antigen to form induce immune complexes which deposit in the vessel, leading to the reactions. Then T cell mediated disease or the type 4 hypersensitivity results from the inflammation caused by cytokines. Okay, and what are these cytokines? Th1, Th17, and all these cells which basically in the end injure the tissue. So it is the enhanced activation of our own immune system. So you need to see there is this immune system which needs to be there to defend us against the pathogens. This immune system should have a feedback or regulatory mechanism so that it can be turned off when not needed. The immune system should be stimulated in the ample amount and should not be stimulated more than it is needed. And the immune system should not affect our own cells. That is, it should have a clear distinction between self and non-self. Okay, otherwise it will lead to autoimmune disorders. Now we're gonna spend little bit more time, so I'm gonna be patient with the questions as always, because I hope that majority of you have registered for the exam, and the best way to prepare for these exams is to first regularly be updated with whatever lectures have been uploaded. Go through them, look at the slides from the live sessions, go to the visual aids, go to the resources listed. You don't have to prepare separately for them. If you follow these 3 steps, you can easily crack the exam. Okay, then I guess we have, so what I'll do is, because this topic we have not covered in our sessions, but it was very well and beautifully covered in the lectures uploaded, but we'll quickly go through the visual aid to remember what class of receptors, type of receptors, and how each subunit signifies different thing. Okay,
naming strategies for these small soluble proteins. Recall that cytokines are used by immune cells to communicate with one another, while chemokines guide the migration and localization of immune cell nomenclature can also get a little confusing, so let's break it down. There are 2 major families of chemokines. Those are the CC family and the CXC family. CC chemokines are named as such because there are 2 adjacent cysteine amino acid residues near the amino terminus of the protein. Each of these residues is represented with the letter C. CXC chemokines also have 2 cysteine residues near the amino terminus, but they are separated by a single variable amino acid, marked by the letter X. Here's a tip. If you find CC or CXC followed by an L, for example, CCL2, the L stands for ligand. CCL2 is therefore a chemokine. If CC or CXC is followed by an R, the R stands for receptor, and you know that this protein is a receptor for either a CC or CXC chemokine. Let's look at some CC chemokines. These tend to induce migration of lymphocytes and monocytes. CCL2, or MCP1, promotes Th2 immunity and release of histamine by basophils. CCL3, or MIP1 alpha, recruits monocytes, macrophages, and neutrophils, and promotes Th1 immunity. CCL4, or MIP1 beta, recruits monocytes and natural killer cells. CCL5, or RANTES, recruits eosinophils, T cells, and basophils to infection sites, and can also activate natural killer cells. Then there are CXC chemokines. These tend to promote neutrophil migration, and they include the following. CXCL8, also known as IL-8, recruits neutrophils to infected tissues. CXCL7 is released from activated platelets. This chemokine activates neutrophils and promotes angiogenesis, which is the growth of new blood vessels. This is especially important following injury and during tissue repair. CXCL1, CXCL2 and CXCL3 are similar to CXCL7. They promote angiogenesis, activate neutrophils, and stimulate fibroblast proliferation. The difference is that these chemokines are released by endothelial cells, fibroblasts, and monocytes instead of platelets. As we mentioned before, chemokines are also very important for directing lymphocytes to where they need to go in the lymph node and other lymphoid tissues. CCL21 is secreted by stromal cells in the lymphoid tissue to recruit dendritic cells. CCL18 and CCL19 are then secreted by dendritic cells, which recruit T and B cells to the lymph node. And CXCL13 is secreted by follicular dendritic cells to recruit B cells to the B cell zone by binding.
This was the extra information because it was not part of our slides. So next question is, I think we have done this, so let me be, yes, we can start from here. Yeah, so the STAT dimer binds to blank element on the DNA, which leads to expression of the interferon stimulated genes. Fill in the blanks of this. It binds to which element of the DNA? Is it ISRE, interferon sensitive response element, interferon controlled rescue elements, interferon dependent rescue elements, or both B or C? You guys have 30 seconds to answer this, and then we'll look into the results. You can put your answers in the chat box. Can you give an idea on the pattern of the question paper? What type of questions to expect? We can discuss this after the question, Asmita. Okay. So during the beginning of this class, I discussed that I want to cover more questions today. So we are going a little bit faster. So the answer is ISRE, that is interferon sensitive response element. We'll quickly see the whole process, like the main thing which I want you guys to focus on.
Are you a work?
Talking about JAK-STAT signaling pathway. This is a very important cell signaling pathway which is important in context of development, physiology, and in terms of disease. So JAK-STAT signaling pathway begins with the cell surface receptor, which are most commonly the cytokine receptors. So cytokine binds to these receptors, leading to its dimerization. So these are the membrane surface events. So these receptors bind to JAK or Janus kinase. So these are kinase enzymes which obviously phosphorylate downstream targets. The first thing the JAKs phosphorylate is the receptor itself. So the receptor phosphorylation is done by JAK. And this is kind of similar but also different from the receptor tyrosine kinase. In the receptor tyrosine kinase, the kinase activity was intrinsic. But in case of JAK-STAT signaling pathway, the kinase activity was not intrinsic to the receptor. The JAK are soluble proteins which associates with the activated receptor and phosphorylate the receptor. So this is the different part. Now, after phosphorylation of the receptor, specific signal transduction proteins known as STATs can bind to these phosphorylated receptors. Now, once STAT binds to these phosphorylated receptor, JAK can further phosphorylate these STAT molecules. STAT stands for signal transducer and activator of transcription. So more or less it's a transcription factor. So phosphorylated STAT can dimerize, either homodimerize or heterodimerize, and eventually translocate into the nucleus. Thereby, it can bind to specific regions known as cytokine response elements. After binding to cytokine response elements, it would recruit RNA polymerase and eventually lead to activation of many genes. And these gene categories would be described in subsequent slides. So the question is, the JAK-STAT signaling formula is same for all the cells. So where does heterogeneity occurs? So the heterogeneity occurs based on the type of JAK used or the type of STAT is involved in the signaling. There are different type of JAKs and STATs that are downstream to different signaling pathway. For example, if you look at this particular slide, you can see IL-4 receptor binds to JAK1 and JAK3. In contrast, interferon gamma receptor binds to JAK1 and JAK2. And they also activate different STAT, whereas where you can see IL-4 activates STAT6, the interferon receptor gamma activates actually STAT1 homodimers. So obviously, these heterogeneous outcomes lead to the differences in cellular responses. So now let's talk about the termination of JAK-STAT signaling pathway, because initiation of any signaling pathway is important, but the termination is more important to overcome the risk of uncontrolled signaling. So there are particular different strategies by which the JAK-STAT signaling pathway can be terminated. There are specific phosphatases known as protein tyrosine phosphatases that can chop off the phosphate group from the receptor and rendering this particular signaling pathway inactive. There are other strategies like receptor-mediated endocytosis of the receptors. So that reduce the number of receptors available for binding to the signal. Also, there could be other proteins which can possibly degrade the JAK or STAT or even the receptor. So these are known as suppressor of cytokine signaling protein or SOCS proteins that can potentially degrade the receptor and degrade the JAK with combination of other molecules. Now let's talk about the JAK-STAT pathway in context of interferon signaling. So we have some understanding about the immunology as well. So interferons are alarming molecules that are secreted by a viral infected cells. So these interferon molecules are recepted by specific receptors on the neighboring cell, and that gives the neighboring cell time and the necessary proteins to counter a viral attack. So interferon signaling is mediated by JAK-STAT. So interferon receptor alpha and beta or gamma receptors bind to specific interferons, and they activate JAK-STAT signaling pathway. And note that here there are specific JAKs which are downstream to each of these signaling receptors. Ultimately, they bind to cytokine response elements and give rise to genes. But the question is, which genes? So let us break that down. So first of all, one of the genes that is getting activated is protein kinase R. So ultimately, protein kinase R enzyme is produced. This particular enzyme actually interacts with the viral DNA and eventually inhibits its further downstream processes. Also, protein kinase R can phosphorylated elongation factor 2A, which blocks the translation. So if the viral genes cannot be transcribed, cannot be translated, virus would eventually die. So these are some antiviral responses. Similarly, there are other molecules such as oligo-A synthetase. This particular molecule processes viral mRNA in such a way that they are more prone to degradation. Another protein known as MX protein, which oligomerizes and inhibits the viral particle assembly. So various aspects of viral life processes are hindered by this particular gene products. This is how JAK-STAT signaling ensures interferon response is augmented. Now let's talk about the JAK-STAT signaling pathway as a therapeutic target. So there are specific...
Okay. So now you have your revision of this. Coming to the next question. Yeah. So oligodendylate synthase, that is OAS, activates RNase L by the production of this. If you have gone through the lectures, you'll be like, you can easily answer this question. You have 20 to 30 seconds, I guess. Yeah, you can put your answers in the chat box. So the right answer is 2,5-oligodendylate. Go back to the lectures. I'm not going to discuss this at least today. We can have a doubt clearing session maybe in the next week if you guys request. Otherwise, yeah, you can always go back to the lectures, and everything which has been discussed in the lectures, chances are a conceptual question will come from there only. So for example, the question which I asked, that if you have a basic molecular weight of an antibody, right, can you distinguish whether it's an IgA, IgG or IgM, right? So that's a conceptual question. If you know the basic concept, you can apply it. Okay, so those type of questions you can expect. The next question for today is, a Rh negative mother may develop a hypersensitivity reaction if she comes in contact with Rh positive fetal blood during her second pregnancy with Rh positive fetus. This is an example of type 1, type 2, type 3 or type 4 hypersensitivity. So the right answer is type 2 hypersensitivity. So these things are, again, I know I am being repetitive, but have been discussed there for your clarification, so that you have a visual representation of these incidences. I'm using the visual aid. It will, like, it basically helps in better remembering things.
Explore how data science.
Hypersensitivity refers to abnormal reactions of the immune system against certain antigens. It includes exaggerated reactions to otherwise harmless environmental antigens, commonly known as allergies, and inappropriate reactions against the body's own antigens, or autoimmune diseases. Reactions can range from a mild rash to damaged organs to fatal anaphylactic shock. There are two principal groups of factors contributing to hypersensitivity: imbalance between effectors and regulators of immune response. In some people, mechanisms that normally moderate the immune system are compromised, causing it to overreact to harmless non-infectious antigens. Self-reactivity of immune cells. During their development in the thymus and bone marrow, T cells and B cells learn to not react to the body's own antigens. Self-reactive cells are normally eliminated, but in some people, some of these cells escape and may attack their own tissues once activated. Hypersensitivity reactions only occur in pre-sensitized individuals. Patients must have had a previous contact with the antigen, which produced no symptoms, but during which the body had started making antibodies or activated immune cells that may cause symptoms in subsequent exposures to the same antigen. Hypersensitivity is classified into 4 types based on mechanisms of action. In type 1 hypersensitivity, a previous exposure to the antigen results in production of a class of antibodies called IgE. IgE molecules bind to their receptors on the surface of mast cells and basophils. Upon re-exposure to the same antigen, or sometimes a similar antigen, the antigen binds to adjacent IgE molecules, bringing their receptors together, triggering a signaling cascade that induces the release of histamine and other inflammatory chemicals. These chemicals cause dilation of blood vessels, smooth muscle spasms, and are responsible for symptoms such as edema, rash, difficulty breathing due to bronchospasm, abdominal cramping, vomiting, and diarrhea. The reactions are immediate within minutes of contact with the antigen and can range from mild to severe. Severe reactions may lead to anaphylactic shock, a life-threatening condition in which blood pressure drops and airways narrow to a dangerous level. Most allergies are type 1 hypersensitivity reactions. In type 2 hypersensitivity, previously formed IgG or IgM antibodies bind to antigens on the surface of a particular cell type. Antibody binding marks the cells for destruction, either by the complement system or phagocytosis. The antibodies may also interfere with normal functions of the cells without killing them. Type 2 is at the basis of many autoimmune diseases, where the body produces antibodies to destroy its own cells. Another example is hemolytic disease of the newborn, where maternal antibodies bind to D antigen on the surface of fetal red blood cells and destroy them. Type 3 hypersensitivity reactions are also mediated by IgM or IgG, but in this case, the antibodies bind to free-floating antigens, forming antibody-antigen complexes. The complement system is activated and inflammation results, causing damage to the affected tissue. A typical example is serum sickness, induced by a large amount of antigens in the blood. Immune complexes are deposited in the walls of blood vessels, triggering their inflammation or vasculitis. Type 4 hypersensitivity is a delayed reaction mediated by T cells. Pre-sensitized T cells are produced during a previous contact with the antigen. Upon re-exposure to the same antigen, T helper cells release inflammatory cytokines, while T killers induce cytotoxic reactions. Typical examples are allergic reactions to substances that come into direct contact with the skin, known as contact dermatitis. Type 4 is also the basis of the tuberculosis skin test.
Release of vasoactive amines.
Have you noticed how you keep delaying the things that...
Release of vasoactive amines is commonly observed in IgE mediated type 2 hypersensitivity, IgE mediated type 1 hypersensitivity, IgE mediated type 4 hypersensitivity, or IgM mediated type 2 hypersensitivity. So you guys have 20 seconds to put your answer in the chat box. The right answer is IgE mediated. The right answer is IgE mediated type 1 hypersensitivity. Which of the following events are associated with release of histamines from the mast cells? Is it cross-linking of IgE with the allergen? Phosphorylation of phospholipase C by protein tyrosine kinase? Protein kinase C and Ca2 plus ions help in microtubule contraction, or all of the above? You guys have 20 seconds to put your answers in the chat box. Good. The right answer is D, that is all of the above, and you can obviously refer to this visual aid. Which of the following is a T cell mediated hypersensitivity? Now, as we have just seen the visual aid, you guys will be able to answer this very quickly. Is it type 1, type 2, type 3, or type 4 hypersensitivity? The right answer is type D, that is type 4. Answer is option D, that is type 4 hypersensitivity. Antibodies which are injected to the Rh negative mother to counter erythroblastosis fetalis are called Rogum anti-isotypic antibody, anti-idiotypic antibody, or IgE. The correct answer is Rogam. You can always, again, all these things were covered in pretty much good details. Okay. The term xenoimmunity refers to immune response to foreign antigen, immune response to self antigen, immune response to transplanted organ, or immune response to commensal microbiota. Even if you don't know the right answer, you can always try to understand the meaning of its word from the components through which it is made. So in biology, xeno refers to something. The right answer is immune response to commensal microbiota. So xenoimmunity refers to the immune response against foreign antigen or tissue from a different species, such as in the context of xenotransplantation, where the recipient's immune system reacts to the transplanted organs or tissues of an animal donor. The concept of autoimmunity was first described by this. Yep. Obviously, we don't need too much time in this. It is Paul Ehrlich, and you can refer to this visual aid where the whole, like, the history of what he did is very beautifully represented. Which of these is not an autoimmune disease? Type 1 diabetes, rheumatoid arthritis, AIDS, or Graves' disease? Obviously, it is AIDS, right? Which of the following is a non-organ specific autoimmune disease? Crohn's disease, systemic lupus erythematosus, psoriasis, or multiple sclerosis? The correct answer is systemic lupus erythematosus. If you refer to this link, beautifully and very well structured, this video will let you know what exactly is SLE. The principal mechanisms of peripheral tolerance include suppression of Treg cells, energy, activation induced cell death, or all of the above. The answer is all of the above. Which of the following option is the mechanism for induction of immune tolerance? Central energy, peripheral energy, clonal energy, or all of the above? The answer is all of the above. So these things, according to me, you can always go back to the books. They'll give you better idea about these concepts. Okay. The function of autoimmune regulator gene is to inhibit T cell development, help T cell to proliferate, promote self expression of tissue specific antigen for T cells to recognize the self, or none of the above. The right answer is promote self-expression of tissue specific antigens for T cells to recognize the self. After exiting thymus, mature T cells are subjected to the secondary selection, where the majority of self-activated T cells are deleted or rendered energetic. The process is known as central energy, peripheral energy, clonal energy, or all of the above. The correct answer is peripheral energy, and we're gonna see because it's a repetitive thing, right? And yes, just give me 1 minute. Why we want to focus on this more? Because again, Nobel Prize, right?
In the previous video, we studied 3 main mechanisms of central B cell tolerance. autoreactive B cells may survive through receptor editing, undergo clonal deletion, or become anergic. But B cells do not encounter all the self-antigens in the bone marrow. Once B cells start circulating, they come in contact with tissue-specific cell surface proteins, secreted proteins, etc. Thus, autoreactive B cells that escape the central B tolerance now enter peripheral circulation. Let's find out how peripheral tolerance prevent activation of such B cells when they encounter their specific antigens. To understand peripheral B cell tolerance, we should be familiar with the mechanisms of B cell activation. Let's recall B cell activation mechanisms in brief. B cell activation is of two types, and this depends on the type of antigens encountered by the B cells. T-independent B cell activation takes place when the antigen encountered by B cell is multivalent. The first signal for B cell activation is generated by clustering or cross-linking of B cell receptors on binding to multivalent antigen. The second and the final signal for B cell activation is generated by danger signals such as PAMPs associated with that antigen. T-dependent B cell activation takes place when the antigen is not multivalent. For example, protein antigens. In this case, activation of B cell takes place by a three-signal process. Signal 1 is derived from the antigen recognition and bind to B cell receptors of the B cell. Once signal 1 is received by the B cell now interacts with a T helper cell. Note that this T helper cell is also activated by the same antigen to which B cell binds. The CD40 receptor of cells binds to CD40 ligand of T helper cells. This generates the second signal for B cell activation. Finally, the third signal for B cell activation is provided by cytokines secreted by T helper cell. This results in the proliferation and differentiation of B cells into antibody-secreting plasma cells. Now suppose these are the B cells that migrated to periphery from the bone marrow. If B cells bind very strongly to a multivalent self-antigen, it receives a signal to undergo apoptosis. And if there is a weak interaction or no interaction, then such B cells mature further in the peripheral lymphoid organs. The main mechanism of peripheral B cell tolerance is energy. Suppose this is an autoreactive B cell in the periphery. It recognizes and binds its specific self-antigen. Now, as we said, B cell activation requires T cell help. This also means that the T helper cell must be autoreactive to the same self-antigen which autoreactive B cell encounters. But autoreactive T cells are also eliminated in central lymphoid organs by mechanisms of central tolerance. Therefore, without T cell help, such autoreactive B cells will not be able to receive the second signal and consequently the third signal for its complete activation. In the absence of signal 2 and signal 3, autoreactive B cells become unresponsive. They become anergic B cells. These anergic B cells eventually die by apoptosis. Thus, the main mechanism of peripheral B cell tolerance is energy followed by apoptosis. In the next video lecture, we
The autoimmune thyroid disorder with the presence of anti-TSH receptor antibodies suggestive of Hashimoto, Graves, Crohn's or psoriasis. The correct answer is Graves' disease. Human beings who lack AIRE are known to develop Crohn's disease, multiple sclerosis, psoriasis, or autoimmune polyendocrinopathy, candidiasis, ectodermal dystrophy. It's D. So even if you don't know exactly, but since if you guys are paying attention to the questions, you would have been able to answer this. By an autoimmune response can be detected after a myocardial infarction. B cells produce antibodies specific for DNA that leads to autoimmune response. Intracellular antigen that are not normally recognized by lymphocytes are released as a result of massive tissue injury that leads to autoimmune response. T cells fail to recognize foreign antigens, or all of the above. So the correct answer is B, and this was discussed very nicely in the lectures. The rheumatoid factor developed in rheumatoid arthritis is anti-IgG antibody, anti-IgM antibody, IgE antibody, or anti-Ig antibody. The correct answer is anti-IgG antibody. The SLE, which one of the following receptors involved in activating autoreactive B cells, is TLR4, 9, NLR, or CLR. It's TLR9. So if you go back to that visual aid, this question will be clear. Immunologically privileged sites of human body does not include brain, eyes, testes, or liver. It's liver. Immunologically privileged sites are distinct as effector T cells are highly active in these sites. A population of memory B cells always remain in these sites. Antigen placed in these sites do not elicit destructive immune responses, or antibodies specific for antigen in these sites do not mediate any effector function. As the name suggests, that it should not have any reaction, right? So antigens placed in these sites do not elicit destructive immune response. Recognition of self-antigen in the peripheral immune tissue in presence of TGF-beta, but in absence of pro-inflammatory cytokines, develop a T cell into natural Treg cells, induced Treg cells, effector T cells, or memory T cells. The answer is induced Treg cells. Inflammatory bowel disease is an example of autoimmune disease where immune response acts against the intestinal epithelial cells, pancreatic cells, gut microbiota, or all of the above. It's in the name, it is the gut microbiota. Experimental autoimmune encephalomyelitis causes inflammation in the brain mediated by Th1 cells specific for acetylcholine receptor, Th2 cells specific for glial cells, Th1 and Th17 cells specific for myelin basic proteins, or Th9 cells specific for astrocytes. It's Th1 and Th17 cells specific for myelin basic proteins. During the manufacturing of vaccine, very small amount of other components such as adjuvant, preservatives, and stabilizers are added along with the antigen to prevent contamination and increase the immune response. All are commonly called adjuvant in the vaccine except potassium aluminium sulfate, formaldehyde, and last aluminium hydroxide. This was covered in this week's lecture, and the answer is formaldehyde. Choose the correct statement about Sabin polio vaccine. So first is, it consists of 3 attenuated strains of poliovirus. It consists of 3 inactivated strains of poliovirus. It is administered orally. It induces production of secretory IgA in the intestine. It requires booster doses. It requires single immunizing dose. So the correct answer is, it consists of 3 attenuated strains of poliovirus. It is administered orally. It induces production of secretory IgA in the intestine, and it requires booster dose. Monoclonal antibodies are used as therapeutic agents because they can specifically target aberrant cells, but there is a major disadvantage. Monoclonal antibodies raised in mouse are expected to develop an immune response when injected to humans. To prevent its difficulty, monoclonal antibodies are humanized by replacing Cl and Ch regions of MAB with that obtained from a human IgG, by insertion of mouse MAB complementary determining regions into human IgG, by insertion of human IgG complementary determining regions onto the mouse monoclonal antibody, replacing Cl and Ch region of human IgG with that obtained from a mouse antibody. The correct answer is by insertion of mouse monoclonal antibody complementary determining regions, because they are the ones which are determining the antigen, right, on human IgG antibody. Immunotoxins are antitoxins, antibody for specific antigen tagged with toxin, formaldehyde inactivated toxin, low immunogenic toxin, or inhibitory chain of bacterial toxin that can induce antibody secretion. The correct answer is antibody for specific antigen tagged with the toxins. Maternal antibodies acquired transplacentally during fetal life and by the intestinal absorption of colostral IgGs are an example of natural active immunization, natural passive immunization, artificial active immunization, or artificial passive immunization. It is natural passive immunization. It is coming from the breast milk. Toxoid vaccines are prepared by inactivating bacterial toxins by treating them with formalin. One such example of toxoid vaccine is typhoid, cholera, tetanus, tuberculosis. The correct answer is tetanus. The conjugated vaccine of Haemophilus influenza type B, which causes bacterial meningitis in children under 5 years of age, is prepared by linking a type B capsular polysaccharide to another type of B capsular polysaccharide, a protein carrier, a lipopolysaccharide carrier, or a carbohydrate carrier. It's a protein carrier. Most of the time the carrier will be the protein only. The adjuvant which is approved by the FDA in the United States for use in market human vaccine is Freund's complete adjuvant, bacterial polysaccharide, alum, or MF59. It's the simplest thing which we have, it's the alum. Edward Jenner made the first smallpox vaccine by performing an unethical experiment. The material used by him for inoculation was a carrier vaccine, a subunit vaccine, an inactivated virus vaccine, a live attenuated virus vaccine. It was a live attenuated virus vaccine. For successful vaccine discovery, viruses are attenuated by passage of live viruses from cell to cell and search for less virulent mutant and use low temperature to select cold adapted mutants. passage of live viruses from cell to cell and search for less virulent mutant and use high temperature to select heat adapted mutants. Direct random mutagenesis to create a less virulent mutant. Formaldehyde and several alkylating agents. The answer is A. Passage of live viruses from cell to cell and search for less virulent mutant and using low temperatures to select cold adapted mutants. This comes to the end of all the questions which we had to discuss. And so the question which you guys asked, that what sort of questions you can expect in your paper, so it is not in my hand. It completely depends upon the professor who is making the paper, but most of the time the questions are combinations of multiple choice questions along with like one word or one sentence questions. This information you can get from the NPTEL page itself, where you will have a description of what sort of questions come. But rest assured, so in your current assignments, so you have already encountered more than 120 questions on your own, right? And if you're attending live sessions, then we have discussed more than like 150 questions. So that comes up to total 250 questions for one subject you have already covered, like on major topics. So I'll say, if you just look at, try to find out the patterns in these questions, it will be easy for you to prepare. But again, the mantra is, if you have been consistent with the weekly lectures and in the live sessions you have attended, you have used the visual aids, you have used the examples, you have used the resources, you don't actually have to sit separately and prepare for it. Okay, so just go back to those lectures as with a mindset of revising them, not learning new thing, but with the mindset of repeating it or revising it. And remember, as I've always told, immunology is like a story. So try to make the story when the microbe enters, what happens to it, and by this way it will be easy for you to learn as well as do well in your exams. Okay, so with this, thank you so much for being very patient with me and being regular in the class. I wish you all the best for your exam and even like for your college exams as well. So yeah, I hope you enjoyed the subject. Okay, bye bye.